So again, thank you for being here, and let me introduce our speaker. Today, Professor Sepideh Sadaghiani will talk about her research on neural communication. She's an assistant professor in the cognitive neuroscience program area of the psychology department here at U of I, and also she is, of course, a member of the neuroscience graduate program. She directs the Connect lab at the Beckman Institute, where she is a full-time faculty member. She earned the PhD in Neural and Behavioral Sciences from the Max Planck Graduate School and did a postdoc at UC Berkeley and at Stanford University. As you can see, and as you will hear today, she investigates the role of neural connectivity and spontaneous brain activity in cognitive control and behavior through a multimodal lens. She's been funded by NIMH and NIH. And as you can see, her title is the functional connectome across spatiotemporal scales. So let's give a warm welcome to Professor Sadaghiani, and Sepideh, take it away. Thank you for being here.
Thanks so much. Thanks. It's a pleasure to see people from across departments here. And I really hope that I'll give enough introduction for everybody who's outside the brain sciences or this particular area of the brain sciences to be able to follow hopefully some of the things I will say. Are you able to see my slides right now?
Yes.
Great, thank you. The topic requires to become more and more technical towards the end of the talk, so I apologize ahead of time. Please feel free to post your questions in the chat if things are unclear, and I can go back to those slides and give more detail. I wanted to start, okay. Well, I do want to start, there we go, with my thank you slide to be sure that I give enough time for credit to everybody in the Connect lab, specifically those from whom I'm going to be presenting data today, as well as those who spend a lot of time right now at the Beckman acquiring concurrent EEG and fMRI data that will allow us to continue this line of work. Ryan Larson, as well as everybody at the BIC who is really making this multimodal work happening. I'm going to also show a lot of data that we have received through collaborations externally. With that, I will start. So the introductory part is really about the organization of functional connectivity in the brain as observed with fMRI. This is the primary method with which the so-called functional connectome of the human brain has been studied to date. I will then move on to show that electrophysiological modalities such as EEG can likewise be used to look at the functional connectome, even though a lot of my colleagues in the cognitive neurosciences would not agree that that is readily possible. So I hope to convince you by showing data from our lab and other labs that in fact we can take advantage of electrophysiological measurements for this. And hopefully it will become clear at the end of the talk of why we want to have more than one modality to study the functional connectome. I will then move on to bimodal studies. So studies that concurrently look at EEG and fMRI to compare their spatial organization. This is the so-called static connectome or the time averaged connectome. So just really looking at how the connectivity of the brain is organized across space. And then I will move on to so-called dynamic connectivity or time varying connectivity and what the temporal relationship is between EEG and fMRI connectivity. So just to recap about our quote unquote traditional view of how electrophysiological measures such as EEG, but also MEG relate to hemodynamic based imaging, specifically fMRI. So when there is neural activity in the brain, and if enough neurons are firing synchronously, especially in these really large, strong pyramidal cells that have aligned dendrites, then we get a large enough electrical current and the magnetic field of it that it can actually be measured non-invasively over the scalp using EEG or MEG. But what fMRI does indirectly is really imaging the hemodynamic response to the same neural activity, or at least so the traditional view goes, with the help, for example, of glial cells there is a dilation of the vasculature, bringing fresh blood into the area where the neurons are active. And the ensuing change of the local magnetic field can then be measured with MRI. So the point here is that in our most traditional view, the same kind of brain activity can be measured with EEG, which is a real-time method. And so it's very good to understand when things are happening. And the same neural activity then also shows up in fMRI. And because fMRI has very good spatial resolution, we can understand where things are happening. But of course, the fMRI signal is very sluggish in time. So it peaks about 5 to 6 seconds after the actual neural activity, and it's very wide also in time. And so it's really a low pass kind of filter, temporal smoothing of the neural activity. So to summarize this, the idea is that there is neural activity that can be imaged with different temporal and spatial qualities in both EEG and fMRI, but then also, of course, there is contributions of noise that are resulting in parts of the EEG signal not being shared with fMRI, and also noise in fMRI, obviously resulting in some kind of discordance potentially between measurements in these two modalities. All right, so let me come to the time averaged or static functional connectome as it's traditionally studied with functional MRI. So I'll first start with this positron emission tomography image. Actually, this is not fMRI, but it images the glucose consumption in the upper body. And you can see that the brain is extremely hungry for energy, much more so than even other organs that are constantly active. And in fact, the brain consumes 20% of the body's energy irrespective of what we're doing. We can be sleeping, we can be exercising or thinking hard. It really doesn't matter. We're always extremely active as far as neural processes are concerned. And these neural processes that are continuously ongoing have actually functional consequences. So imagine I'm playing a sound, and then it basically goes through our sensory system, and it arrives in the brain and results in some kind of an electric wave. But if I do the same thing again, then the wave looks slightly different on every trial, essentially, of this sound. And so typically what we're used to see is average responses, in this case, average electrophysiological response. But the reality is that if you look at the individual responses to the same sound, they're actually quite different from one occurrence to the other. And there is a lot of background fluctuations that are actually looked quantitatively. They're actually larger in amplitude and also just in the sheer amount compared to the evoked signal. So most of brain activity is actually intrinsic, not evoked by external stimuli. And it actually has an impact on how the brain is processing incoming signals, like the sound. And so one of the first functional MRI studies that looked at intrinsic activity was quite early on by Bharat Biswal. Here I'm showing a horizontal slice through the brain, so it would look something like this, measured with fMRI. And here you see a finger tapping task. So the subject was asked to actually tap their fingers. And what you can see is hemodynamic signal changes exactly where you would expect them, which is in left and right motor cortex hand areas, as well as supplementary motor areas. But the point of this study was actually that when the subject is doing absolutely nothing in a task-free resting state, if you look at the spontaneous fluctuations of the background signal, so it's the kind of signals that I just showed between the actual events, for example, this is background fluctuations or that intrinsic activity, that those are highly correlated among the same regions of the motor system. So in this case, Bharat put a voxel in just a single voxel during analysis in the motor area, and then looked at all the other voxels of the brain to see where both signals are correlated with that region. So he found that the same functional network that's active during task is also actually crosstalking. The same regions are communicating during resting state. And so this communication can be quantified simply as Pearson correlation. So imagine these two regions are functionally connected, like the two motor regions that I just showed, for example. They would have a very strong correlation value. In contrast, regions that are not functionally connected at resting state will have a low correlation value. So when you do this kind of correlation-based analysis, you can retrieve the motor system that I just showed. But you can also put your seed voxel, the so-called area to which you're comparing the signal from the other regions of the brain to. And you can look at any part of the brain really and look at what other parts of the brain are communicating with that region, and you get all of these different brain networks. So what's interesting about these networks is they tend to be the same areas that co-activate when we're engaged in tasks. So if we have, for example, an attention demanding task, then regions involved in attention here, spatial attention here shown in dark blue, for example, will be co-active during that task, but those same regions seem to be kind of going hand in hand during resting state. So if we look at the relationship of any region by any region and build these correlation values for all region pairs of the brain, we get something like this, which is called a connectivity matrix, or in other words, the whole brain connectome. So this is what we call a connectome. And if you organize those regions by these so-called canonical brain networks that are shown in different colors here and here, then you get these kind of high correlation areas along the diagonal. The connectome can also be looked at as a graph in physical space or even in so-called topological space. You can basically think of this graph as the brain's social network. So we're more used to thinking about our life as graphs, really. So this would be a social network of a quote-unquote seed person on LinkedIn or maybe Facebook. And this person has connections to a lot of other people, but those people tend to come from different groups. So these would be the different networks, like the motor system or the attention system and so on. So this is the whole brain connectome. One interesting aspect that has been seen in functional MRI is that it doesn't really matter whether the subject is in task-free resting state or is doing any kind of cognitively demanding, behaviorally demanding task. These connectomes seem to be spatially extremely correlated to each other. Strongly correlated, especially if they are averaged across subjects. So in this case, this particular paper is averaging the connectome first across a large number of participants and then comparing the mental conditions to each other. Very high correlation. So it really means that there is something intrinsic, that is something inherent to the human brain, not driven by the external environment, that is really characterizing our connectome. However, there are subtle changes in the connectome. There are subtle time-varying dynamics that actually do have an impact on our behavior. So here I'm just showing a schematic, again, of that sound experiment. Imagine I'm playing just a sound stimulus to the subject at this point, and there is some kind of a brain response to it, in this case an fMRI. And each time I do it, there is a brain response. But if the sound is at threshold detection level, so it's a very faint sound, and the subject sometimes perceives it and sometimes misses it, then I can try to find out whether the connectome's organization right prior to the sound presentation has any impact on behavior. And I wouldn't be telling you this if it didn't. So imagine I'm building the connectome from this time point and then from this time point, and I feed it into a machine learning algorithm, and I'm able to actually predict whether the subject is going to be perceiving the task on the next trial or not. So I can just look at your brain and I will know what you will do next just by looking at the subtle changes in the connectome from moment to moment. So moving on, most of the rest of the talk will be resting state, but I will also have a little bit of task data in there. Okay, so as promised, now I'm moving to electrophysiology. So up until this point, this was pretty standard approaches using fMRI to look at the static connectome. But we have all this other world of methods that allow to look at the brain from a very different viewpoint, a very different lens. So when we come to electrophysiology, most of the connectivity measures are based on the idea that ongoing neural activity, or this intrinsic activity that I was talking about, is actually fundamentally oscillatory in nature, or a large part of it is oscillatory in nature. And so electrophysiology has a lot more information, and it makes it a lot more complicated to look at than fMRI when we want to derive connectivity. So for example, there might be two regions in the brain where oscillations actually lock to each other in terms of their phases. So these two regions, for example, you would imagine that the signal here in red comes from the red brain region, in green from the green brain region, and they have some kind of oscillatory neural activity. So the phases can align, meaning these regions are somehow basically showing some level of connectivity to each other. They are aligning their processing windows to each other. However, alternatively, the relationship between two brain regions can also be defined by the magnitude of oscillatory activity. We call that amplitude coupling. So the green region, again, also has changes in the amount and the strength to which that particular oscillation is expressed. And another brain region may also amp up that same oscillation at the same time. So this would show amplitude coupling between these two brain regions. We can, of course, have both at the same time, phase and amplitude coupling. And there are higher order measures that are cross frequency, but I don't want to make it even more complicated. The other point of complication, or the richness really of electrophysiological activity, is that in contrast to bolt signal, it crosses 4 orders of magnitude. So EEG is meaningful all the way from very, very slow, or we call them infra-slow frequency ranges, all the way to the so-called canonical oscillation ranges, which are up to 100 hertz and above. So there are different bands of oscillations, which we call delta, and so on, all the way up to high gamma. So I'll come to some of those band-specific connectivity measures in a little bit. One major source of connectivity noise, one major source of noise that matters heavily for connectivity in EEG is signal leakage. So the problem is that the electrode typically in non-invasive recordings is sitting over the scalp, and the electricity that comes from the neurons, from the neuronal sources of the brain, are actually spreading over space. So we might be seeing similar signals in different electrodes, but that doesn't mean that they're actually reflecting two different neuronal sources from the brain. And if we see that those signals actually couple to each other, it doesn't mean that two brain regions are coupled to each other, but it may just mean that electrical signal from one and the same neural population has been spreading across different electrodes. This is the region, as I mentioned, this is the reason, as I mentioned, for why most neuroscientists may be hesitant in thinking of EEG as a good measure, or a sufficiently good measure to look at whole brain connectomes. So this is a schematic of how it can be achieved in EEG, however, similarly in MEG. We would go from the raw EEG signal of each electrode to a bandpass filtered signal. So these are the canonical oscillations that I was talking about in the different frequency ranges. And then this signal needs to be source localized. So as I mentioned, what we measure in the individual electrodes doesn't reflect the signal directly from under the electrode. So we have to somehow mathematically solve an inverse problem and go from the sensors to brain source space. And these brain sources are then averaged across so-called parcellations, or different parcels of the brain, similar to what I showed earlier for fMRI. We can then, for every parcel, look at the oscillation and its phase, or we look at its magnitude for amplitude coupling. So we can do pairwise phase coupling or amplitude coupling, derive functional connectomes similar to the ones that I showed for fMRI, or we can use graph-related measures on these functional connectomes. So some Conservative steps need to be taken here. We need, for example, to disregard, unfortunately, a large part of our signal, or orthogonalize the signals. I'm not going to go into the details of this, but just to show you that there have been recent methodological advances to really make sure that what we're looking at in terms of connectivity in source space is not actually driven by signal leakage. Okay, so here's one of the early examples from MEG that we can retrieve some of these canonical networks during resting state, just looking at spontaneous fluctuations in MEG source space. Similarly, other methods have been applied. This is temporal ICA to actually show side by side connectomes derived from MEG here in the bottom and from fMRI in the top. And you can see this for various different networks. There's always fMRI on the top and MEG on the bottom. There is an astonishing spatial similarity between these sets of networks across the two modalities. So this really should give us a good support that we actually can use electrophysiological measures to derive this kind of ongoing activity, this intrinsic activity and its spatial dependencies, which really derive the connectome. I'm now going to move to some of our data where I will be focusing on intracranial EEG at first, in order to establish that even in the absence of source leakage, we can actually observe some of the core features of the functional connectome. So intracranial EEG is recorded by placing the electrode, for example, right under the dura. The dura is a leather-like skin that is under the skull. You can see this here in operation. These operations are done in patients who suffer from epilepsy and don't respond very well to medication. So these are our traditional patients from which we can get invasive measurements in humans. So the electrodes are directly placed on the cortical surface. This is called electrocorticography. We also have some data with depth electrodes that are actually going deep into the cortex. Okay, so this is how the coverage might look like in 3 example subjects. Of course, unfortunately, we won't have whole brain coverage. So that's the big shortcoming of intracranial data. But the advantage, of course, is that we really can establish some of the core points first in the absence of signal leakage before we then move to scalp data. Nevertheless, in spite of this limited coverage, we are actually crossing different lobes here. So we're really looking at connectivity across functionally distinct brain regions. So here, just an example from a single subject. This is a connectivity matrix from whatever electrodes we had available in that patient. And I'm showing activity for the theta band during resting state and an extremely cognitively demanding working memory task. And you see that there is very strong spatial correlation between these two matrices, similar to what I showed earlier for fMRI. This correlation can be all the way up to 0.9 or even higher, depending on the coverage of the patient and depending on the frequency band we look at. So this is the same patient using another frequency band. So extreme similarity here. And now I'm plotting 1 minus those correlation values. So essentially the dissimilarity, where 1 would be the perfect correlation, and this is essentially 1 minus that correlation. So the point of this radar plot really is that the dissimilarity between task-free resting state and any cognitive task is not necessarily bigger than staying within the same mental state. In fact, even comparing something like finger movements to speech or to a working memory task really doesn't make that intrinsic stable organization of the connectome to differ much across these mental states. So we're here confirming that what is known from fMRI can also be observed in really fast oscillation-based connectivity. Okay, so let me summarize this part before we move to the bimodal data. Most of brain activity is actually intrinsic and not evoked externally. And this intrinsic activity is not white noise. Most of our neuroscience methods actually treat it as if it were white noise. We average it away, or we in our regression models, it all goes into the error term. But it actually is not unstructured. It's very structured, and the structure is very stable over time. This organization can be meaningfully studied using EEG, both scalp level as well as intracranially. And the stability of mental states can be observed in both fMRI and in EEG. So I'm now coming to multimodal studies. I will first cover those time-averaged or so-called static connectomes. So this is data by my former postdoc, where we used concurrent recordings of EEG and fMRI. We essentially look at connectivity within the same parcellation of the brain, and therefore we get a connectome, a time-averaged connectome for fMRI, and we get one connectome for each one of the canonical frequency bands of EEG. In this case, he used the coherence measure, and as I said earlier, throwing away the non-zero contribution, the zero lag contributions to it, so imaginary coherence. So the details here are not important, just realizing that we have a connectome that can be spatially compared to fMRI because it's essentially representing the same brain regions. And here I'm showing you the element-wise, so connection-wise correlation between fMRI and EEG. So if for every one of these region pairs, which we call connections or edges, we assess the connectivity value in fMRI and the connectivity value in EEG, and then simply plot them against each other, we get a fairly decent moderate size relationship here. So this really should give us assurance that even scalp EEG can be used to derive meaningful functional connectomes. What's also really interesting, I think, for those who are skeptical about EEG connectomes, is the comparison to structural connectomes. So as you hopefully know, we have colleagues here that are really the experts in structural connectivity. So I don't dare to go much into detail other than telling you that non-invasively we can measure diffusion-based signals, and then can track those to build essentially these white matter tracts structural connectivity non-invasively. And so when we look at the relationship of fMRI and structural connectivity, we find a decent sized relationship between the strength of connection of every connection in fMRI to the respective connection and structure. So don't mind the directionality, that this is negative. This is because of the particular measure that was used in the structural connectivity. But what's really striking, I think, is that at least for the, depending on the band, the correlation can be at least as good or even better for EEG connectivity against structure than for fMRI connectivity against structure. So in early days, this relationship of fMRI to structural connectivity was taken to give peace of mind that what we're looking at in fMRI resting state is actually not noise driven by vasculature or whatever, but that it's really, reflecting some kind of aspects of the connectome of the brain. With that same logic applied, then this data really should convince us that EEG can be used meaningfully in source space to look at the connectome. All right, so when we derive these correlation values between fMRI and EEG, we can actually ask to what degree that the strength of that relationship is actually a matter of the amount of data or the quality of data or the hardware that we're using. So in a recent work by Jonathan Biazich, what we did is actually directly quantitatively comparing that spatial correlation value between the connectivity matrix in fMRI and the connectivity matrix in EEG at anything between 1.5 Tesla to 7 Tesla, and anything between 64 to 256 channels in the EEG. These are different EEG manufacturers and various different subjects from different countries and different number of subjects and so on. And no matter what we do, we always end up with this range of 0.3 correlation between the two modalities. So there seems to be something that might be going over and above just a methodological noise in the two modalities that might be driving this correlation to not reach higher values. And so one aspect is, when I say settings here, I don't mean just hardware. Here's the number of subjects. It seems that depending on the frequency band, anywhere between 7 and 12 or so subjects will give us 99% of the spatial relationship between EEG and fMRI. It doesn't matter how much more data we add. Same thing holds true if we take anything above 5 minutes of recording per subject, we don't get any further improvement. If we take anything above 9 individual subjects, we don't get any further improvement. So there was some kind of a cap there, and that's kind of curious. Another interesting data point, I think, is here a study intracranially by Danny Bassett's group. They're looking at electrocorticography, so those electrodes that are directly on the cortex. What they're doing here is actually pooling the connections that are available across all patients. So this is a patient averaged connectome. And not every patient will have coverage of every edge, but you can, if you have a large enough pool of subjects, around 100 or so they had, then you can build this whole brain connectome and compare that spatially to fMRI connectome average across a different set of subjects. You get a similar size of spatial correlation, and that holds true irrespective of the frequency band that they're looking at. So again, there seems to be something about this number that really might represent something about the brain rather than differences in methodology necessarily, or noise in methodology, I should say. There are differences that are driving this, but not necessarily methodological noise. All right, so this might look complicated, but I hope I can make it really, really simple. So one thing that we asked is, can we take the bimodal data of EEG fMRI, this is scalp EEG and fMRI concurrently, and move beyond correlations. So here, instead of doing a spatial correlation, we actually decomposed the data. So we're using a method called independent component analysis. But for those who are familiar with this method in brain connectomics, typically it's applied on region signal time series. Here we are applying it on connectivity values. So we take the connectome of every subject from fMRI and the connectome of every subject from the EEG and every frequency band. We put it all together, we stack it all together, and then we decompose it. So this data-driven decomposition is going to tell us which spatial patterns of the connectome are co-expressed across subjects in a spatially independent manner. So this is independent component analysis in space. So it gives us spatial independent component traits, so to say. And each one of these traits has an fMRI counterpart and an EEG counterpart, and frequency fingerprints in the EEG. These are the weights that are contributing to these patterns. So we found two very stable patterns that were highly reproducible in two very different data sets. And the first bimodal pattern, the first bimodal trait had these signatures along the diagonal, meaning it's aligning well with these traditional neurocognitive networks, and above chance similarity to the EEG. So the EEG is also somewhat representative of that same intrinsic organization of the connectome. But we found the second component that is co-expressed across, it co-varies across subjects hand in hand, but has spatially nothing to do between the fMRI and the EEG. So this is really curious. how are these things linked when they're spatially completely discordant? So this is another data point I will come back to just in a little bit. And the last few minutes, I will cover concurrent data in EEG and fMRI that are actually time varying. So now we're moving from looking at time average connectomes to so-called time varying connectomes. So time average connectomes, the so-called static connectomes, are the average value of connectivity across the entire data recording. However, it has been shown that if we build these whole brain connectivity matrices for different time points, we actually observe things that are somewhat different. They may sometimes look quite similar to this average connectome, but if we zoom in, at other times it may just look completely different. There are some approaches that suggest that these connectivity patterns might be somewhat discrete, that we can think of them as connectome states. And it has been suggested that these different states actually will represent cognitive modes or cognitive architectures, each of which are actually good for different types of cognitive processes. And the brain really explores them. So the brain is essentially switching through these possible configurations. If you remember the study that I showed very early on, where we played this faint sound for the subject over and over again, it seemed that depending on which one of these states the brain was in right before we presented the sound, the subject was actually more likely to miss or actually hear the sound. So we have evidence to suggest that these connectome states are actually affecting behavior. So this is scalp EEG concurrent fMRI. What we did is what I just showed, namely building a connectome for short periods of time. In this case, we have for windows that are 1 minute wide, stepping every 2 seconds. We had one connectome in fMRI and one in a given EEG band. We then looked at the similarity of the time course of connectivity for each one of these connections separately. So this is a measure that's allowing for nonlinearity. So instead of temporal correlations, we looked at mutual information. The details don't matter here, other than remembering that for every connection of the connectome, we looked at whether the fluctuations in connectivity over time in fMRI are accompanied by similar fluctuations in EEG. So for every connection, I'm showing here the group level statistics. So this is a t-test essentially. You will see that in almost 100% of the edges, as long as we have enough data, this was really long recordings. As long as we have enough data, we see that there is an above chance relationship between the fluctuations of connectivity strength in EEG and fMRI for any EEG frequency band. And if you look at this, this is sort of a circle representation of the connections of the brain. I'm just showing the top 200 connections. So this is only about 10% of the connections. You will see that for all frequency bands, the region pairs that showed a strong relationship between EEG and fMRI connectivity are vastly distributed across the brain. So they're not really local, they're really large patterns. All right. But we wanted to improve on this and ask a slightly different question. Instead of evaluating every single connection of the connectome separately, which is what I just showed, we here asked, can we look at the entire configuration? So that's the full connectivity matrix in EEG and fMRI, and ask whether they are similar. Are they spatially similar at any given moment in time? Because we know that spatial configuration actually changes. I call it here reconfigurations. It's those states, discrete state changes that I was talking about. Do those states co-occur in fMRI and EEG? And here I'm switching to a different data set, and the reason I'm highlighting it is this is intracranial EEG with concurrent fMRI. And this is an absolutely unique data set in the whole world. As far as I know, there is some other groups that are starting to do this, but this is data that was collected partly a long time ago and generously shared with us from University College London, Louis Lemieux and colleagues. It's absolutely unique. We have invasive data from humans at 3 Tesla. In fact, sorry, this is 1.5 Tesla data. We also have the 3 Tesla data, but I'll be showing the 1.5 Tesla data concurrent with EEG from the surgical patients. So this includes both a surface electrocorticography as well as depth electrodes. We also did another improvement. Instead of having those 60-second windows that I just showed, we were able to use a more recent connectivity measure for fMRI that allows us much better temporal resolution and fMRI connectivity. So we're looking at faster dynamics, and we're looking at the whole spatial pattern as opposed to every edge separately. So this is going to be the last data set that I'm showing you. So what we asked is essentially, if we look at the connectivity matrix in fMRI, and then in EEG, here I have 2 different EEG measures, as I mentioned, amplitude coupling and phase coupling can be conceptualized as 2 different measures. For every point in time separately, I'm looking at the spatial correlation between fMRI and EEG. I'm just essentially plotting this here over time. And you can see that at certain times, this correlation reaches rather high values. And at other times, the 2 patterns are really not similar, not much similar at all. So again, this is the method that I just showed you. We are building connectivity matrices of 10-second duration, stepping every 100 milliseconds or so. We're looking at the spatial similarity measured as spatial correlations, Pearson correlations over time. And the area grayed out here is what would have been an appropriate null model. So only these dark black lines that are over and above the null model are actually showing a non-random real similarity between EEG and fMRI. So we applied an alternative data-driven method to extract discrete states, and those are color-coded here in the bottom. So the same state pairs would, for example, this color, if it reoccurs somewhere else, it'll be the same brain state. But this alternative method also confirmed that these time points in which the correlation between the 2 modalities is really high represent certain discrete states of the connectome that seem to co-occur in the intracranial EEG and the concurrent fMRI. However, there is a lot of time points in which we don't see above chance similarity. In fact, if we quantify that, these points rise above the null model only about 10% of the time. And this is intracranial data, so we're not actually worried about signal leakage contributing to this discrepancy. And the last point that I want to emphasize that's actually interesting is, let me skip this. We can come back to this if anybody's interested. I want to make sure I stay on time so you guys have time for questions. So I'm concluding that the cross-modal similarity of the time-averaged connectome is significant in both EEG and fMRI, across EEG and fMRI. However, the similarity is moderate, and it's actually small. I didn't show data for that, but both intracranially as well as for scalp data, the relationship is actually extremely small, 0.1 or 0.15 or so, when we're looking at individual subjects' connectomes as opposed to subject average connectomes, independent of all of the possible hardware settings we have, different brain parcellations that we use, different amount of data within and across subjects. The cross-modal association of connection-wise changes in the connectivity is actually really linked between the 2 modalities. However, when we look at the spatial organization, which I call states, they only meet between EEG and fMRI on very rare occasions. We also have a data set that I don't show here of scalp EEG and fMRI that suggests that the same spatial patterns, so the same kind of states of the connectome, are expressed in fMRI and EEG. They're spatially similar, but they just don't co-occur. They are largely occurring in independent time points because the EEG cycles through them much, much more quickly than fMRI does. So there seems to be evidence that there is some divergence between fMRI connectivity and EEG connectivity that might actually not be driven primarily by methodological issues or noise, but might have some neuronal contributions. So coming back to the schematic, we have neural activity that is reflected in both EEG and fMRI, and we're talking about noise contributing separately to EEG and fMRI and making the two signals similar. However, there might actually also be neural activity that is primarily captured by EEG and not so well represented in fMRI, and vice versa. So one interesting data point here that I actually want to cite directly from Rita Hari and colleagues, she's primarily an MEG researcher, is the following. And I can't really read my screen now, so let me turn this off. All right. MEG, EEG weights strongly the fastest conducting pathways, whereas fMRI probably receives its main contribution from neural ensembles that are connected via slow and thin fibers, thereby apparently reflecting functionally different brain activations. If you're interested, we can discuss for why this would be the case, but it could explain some of the discrepancies that we have seen with respect to connectivity. So I'm closing by this analogy, perhaps, that we have these really thick fibers, like the highways here, that are well myelinated and super fast. And then we have these smaller roads that are rather thin and not so well myelinated and slower. And they are contributing to different amounts to what EEG and MEG is able to see and to what fMRI is able to see. Some of them may actually be running in parallel, like frontage roads that are, accompanying highways along the same routes, but they're really fundamentally different neurons. And you might say, well, why would I care, right? Why would I care about methodology unless I'm doing this kind of research? Well, it tells us something about the brain. It tells us that the brain might be actually a multi-timescale system. And the reason for that is that cognition itself has processes that primarily function at different timescales. And so, again, I would highly recommend Rita Hari's talk. This is from Human Brain Mapping in Rome in 2019. I shrunk that path so you can copy it if you need. She doesn't focus on connectivity. She's generally talking about brain activity, but I think that the concept of connecting different cognitive processing speeds to different neural processing speeds and what can be captured by different brain modalities is beautifully captured in that talk. And with that, I close thanking everybody again for being here and those who actually generated the data. Thank you.
All right, Sepideh, thank you so much. Let's give you a virtual round of applause with our little reaction button in Zoom. So thank you again for a really interesting and well-paced and exciting talk about these different modalities. We do have a couple of questions in the chat, so let's go to those first. Kwanjin is asking about auditory stimuli in a noisy scanner and the region for connectivity being large. Do you want to take the auditory stimulus question first?
Oh, these are two different questions?
Yeah,
okay, I thought I was worried that the one is driving the other.
Oh, sorry.
Yes, I assume the first question relates to that auditory detection experiment. Yes. In fact, we're taking advantage of the scanner as being background noise. The same study had been done about 2 decades earlier using EEG, and they were using tapes back in the day, and they actually had tapes with white noise running in the background. It makes it simpler to have a threshold detection task. So if you look at the visual version of this, also people have things like object recognition over a background of noise. It's kind of just a general approach of looking at signal over noise. So we were really thinking about the background as noise. Now, if you're asking whether that could explain connectome differences at baseline, I don't really think so. The stimuli were, in this case, 20 to 40 seconds apart, because we really wanted these clean baseline sections after the previous hemodynamic response has completely subsided. And the scanner was running, so the repetition time was 1.5 seconds, and so you would think that all of these like sounds would repeat every 1.5 seconds, and at a much faster scale than the connectome that we built over that interstimulus interval. So I don't think that that had any impact on our findings.
Okay, and Kwanjan's second question is, the region for connectivity is large. Oops, and we have more questions that are popping in, so now I have to scroll. Can I scroll up in the chat? I'm not sure. Let me see here. Close. Okay, the region for connectivity is large. Can we assume that the region should be uniform in the signal time course? Maybe, yes. Kwanjan, if you want to ask, and if you want to unmute yourself, you can ask this in more detail too.
Yeah, I mean, there have been approaches to look at the connectome in fMRI on a voxel-wise basis, right? Building those connectivity matrices in really enormous sizes. If you take every one of the cortical 30,000 voxels or whatever. But there are also other approaches. Rather than taking a canonical atlas, you would grow the region itself as by taking into account its the similarity of every voxel with its neighbor in terms of its connectivity to the rest of the brain. So you would parcel the brain into connectivity-wise homogeneous regions. And it seems that all of these approaches, whether you take an anatomical atlas, functionally defined atlases, resting state driven atlases, or these kind of like, atlases that are individually driven by the subject's own connectivity patterns or so, we do seem to see a lot of these points being robust across those aspects. For example, the fact that there is such a thing as a stable connectome over mental states and so on. So what I typically do is try to show supplementary data in an alternative atlas. But when it comes to EEG, you're absolutely right that we don't have that luxury. The EEG is fundamentally limited in terms of the number of regions an atlas can own or have, because if we have a 64-channel EEG, we can't source localize it to a 250 region parcel. That's just like mathematically not going to work out. So for EEG, we are limited to a few pretty large parcel atlases. But again, some of our data has been shown in alternative parcellations.
All right, we have a couple of other questions in the chat. Aaron is asking, for the EEG fMRI studies, are there any surface EEG studies run pre and post the intracranial studies?
Parham can correct me here if I'm wrong. We don't have the scalp EEG in the same recording. Or Parham, did we have the scalp EEG in the same recording? I mean, I know we didn't receive the data, but it exists, right? Or is it offline, scalp EEG with intracranial? I know he's here, I saw him.
Yeah, I'm here. Hi. I think it is offline, so it's not concurrent EEG fMRI.
But we have offline concurrent scalp EEG with intracranial EEG, right?
I'm sorry, can you say that again?
But they do have offline scalp EEG with intracranial.
Yes, same subjects, but offline recording of EEG and fMRI.
Aaron, did that answer your question?
Yeah, Aaron.
Yeah, I mean, probably the follow-up question would just be, there's always this connection between surface EEG, fMRI, and then intracranial EEG. I assume you're just trying to always map that. So just kind of wondering how close you can get to actually doing that.
So, my phone call, Jonathan is also working with this data to get at that exact question. So he's much more, he likes quantitative and more methodological questions. And so he's working with the same data set. As I mentioned, we also have 3 Tesla data from these same subjects, and he will be working concurrent scalp EEG without intracranial EEG, but concurrent with fMRI in the same subjects. And so there is various pieces to this data, and he is going to compare the cross-modal relationship of intracranial to fMRI and scalp to fMRI. That's going to be, I mean, he's actively right now working to answer that question.
All right, thanks. We got two more questions real quick, maybe hopefully real quick. Ava Fisher asks, there are examples where changes in behavior are linked to changes in functional connectivity. How do these findings interact with the fact that regions do appear to be consistently functionally connected? So there's always something going on, but then also changes in the connectivity change are correlated with behavioral changes too.
So do I hear the question right, that how it can concurrently be true that we have very stable connectivity as well as connectivity changes affecting behavior?
Yes.
Yeah, so yeah, that's a very good point. The way I'm trying to conceptualize it is that connectivity overall, its spatial pattern is very stable, but there are the changes, even though subtle, that are there, and they are behaviorally meaningful. That's the way I'm trying to think about it. Yeah, this is the way I'm basically conceptualizing it.
So I have a follow-up question. Does that mean that we each have a brain fingerprint? That if
Yes.
I went into if I got my brain scanned,
Yes.
that like my own
Right. It's
kind of
a big field, actually. It's called connectome fingerprinting. So you're right on point. Yes. And as you might imagine, the individualized fingerprinting literature is mostly driven by fMRI. There is one really nice preprint right now based on MEG of the human connectome data. I'm happy to point you to it if you're interested. But there is people are starting to do this with MEG data because the Human Connectome Project is providing MEG data as well. Most of it is fMRI, and it shows across subjects that cognitive abilities are linked to individuals' static connectome organization. And also, there's also literature showing that the dynamic features of how the connectome reorganizes from moment to moment is also linked to cognitive abilities across subjects. And so in terms of fingerprinting, it has been shown that machine learning algorithms are able to identify you, Patty, based on your data among hundreds of other subjects. They can find you, again, looking at your connectome.
Interesting. All right, one last question from Julia. Aside from fMRI displaying sluggish peaks of 5 to 6 seconds after neural activity, what are other limitations of fMRI while collecting data?
There is a lot of limitations, of course, of fMRI. One particularly interesting one, I think, in the context of connectivity is some data that seems to suggest, so this has to still be, more extensively studied and validated, data from Molly Bright's lab showing that the same regions that tend to co-activate might actually have a correlated vascular response, even in the absence of co-activation. So you would imagine, for example, if the left and right motor cortex are anticipating to become activated in a moment, they are receiving fresh blood at the same time, because we know those regions are going to co-engage. And that's kind of, although evolutionary, it makes a lot of sense, it's also kind of scary, because it would mean that the same connectivity networks would have a co-vasculature response that is over and above the neural correlation. So.
All right, well, thank you so much. We're just over time, so I'm gonna stop the questions for the moment. Again, thank you, Sepideh, for a wonderful talk, and again, virtual hand clapping from the audience. And I would like to remind everybody, first of all, that this seminar is being recorded. We will get Zoom to process it, and then we will have a link available if you would like to download it or share with your colleagues. So let me or Stacy Olson know if you'd like that. And secondly, please join us again for the next installment of the Beckman Institute Director Seminar Series on April 1st. We will feature Professor Sam Tawfik talking about his work on kinetic materials, and Sam is in the Mechanical Science and Engineering Department. Once again, thank you so much, Sepideh, for a wonderful talk, and thank you all for being here today. And we will close the seminar and adjourn. Thank you so much.